Thank you. Welcome, everyone. We're about to start.
Hi, Ash. Good afternoon. This is Lancelot Samuel here, former schoolmate.
Hi, Mr. Samuel. Hi, Mr. Samuel. Nice to see you. Nice to hear from you, sorry.
Okay, all right. Thank you.
Okay, so by my watch, it is 5 p.m. So I think we shall begin now. Hi, and welcome to the National Trust of Trinidad and Tobago's guest lecture, Ancient DNA and the Genetic History of the Caribbean, presented by Dr. Kathrin Nagele. My name is Ashley Morris. I'm a Heritage Preservation and Research Officer here at the National Trust and a PhD candidate in archaeology at Leiden University in the Netherlands. I'm especially pleased to be hosting this very important discourse on the results of almost 30 years of collaborative research. First, the general format and house rules. I'll conduct a brief introduction, then we will listen to Dr. Nagele's pre-recorded presentation, followed by a 20-minute discussion where Dr. Nagele will join us live. Please keep your mics muted for the duration of the presentation, but feel free to use the chat to ask any burning questions you may have. In the discussion, we will invite the audience to participate live or ask questions via the chat function. Please keep your questions or comments brief so we can hear from as many of you as possible in the allotted time. Dr. Nagele will share her email address at the end so you can contact her directly with any other questions that we will cover today. Now, a bit about our organization. The National Trust of Trinidad and Tobago is a membership-based organization established by an Act of Parliament in 1991 to safeguard and promote our nation's tangible patrimony. I cannot tell you how thrilled I am to see so many of our membership here with us this evening. Your continued support in all we do is truly incredible. If for some reason you are not yet a member of our family, please consider joining using the links found down in the chat. And Ty will be so kind as to put those links in the chat for you. I'd like to recognize the members of both the Santa Rosa First Peoples community and the Warao Nation who are present with us this evening. Thank you for being a part of this event, which is only the latest milestone in our continued collaboration. So, archaeological heritage is an important facet of our national patrimony, and the Trust has a long track record of providing a platform for both research and public dialogue on archaeological matters and other related fields. However, we do acknowledge the lack of a cohesive national policy framework for the management of our archaeological heritage. Such frameworks are crucial for research that involves human remains, which can be the most informative and the most sensitive aspect of archaeological research. We hope that this presentation today can be a catalyst for open and inclusive discussions between national authorities and our local First People communities on this much needed legislation. The presentation we hear today, as I said, is a result of over 30 years of mutual collaboration between local and international researchers, included but not limited to Dr. Ari Boomert, the late Peter O'Brien Harris, and Professor Basil Reid, amongst others, many, many others. Dr. Nagele's work involves samples from both the Red House archaeological site and Manzanilla 1 site. I think it's very important to state that both these projects involve substantial community engagement efforts. I'm also especially proud of the regional precedent established during the Red House project, our first people's community was actively involved in the process, which culminated in the reinterment ceremony of 2019, where the treasured remains of our indigenous ancestors were reburied according to rite and custom of our descendant First People community. In keeping with this precedent, we would like to ask for your help in guiding this research to the topics and questions that most interest you, the people of Trinidad and Tobago. I challenge both local and international researchers to consider adopting collaborative, inclusive, and co-creative practices in future scientific research, just like the ones that are used in this research. Now, without further ado, I introduce Dr. Nagele. Kathrin Nagele is currently a postdoctoral researcher at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, where she focuses on studying the population history of the Caribbean and circum-Caribbean region. During her PhD at the Max Planck Institute for the Science of Human History, she gained expertise in retrieving DNA from regions with poor DNA preservation and applies her experience in tropical areas, focusing on the Caribbean and the Pacific. From her focus on marine biology during her undergraduate studies, she has retained her view on oceans as connectors, not barriers. Please join me in welcoming Dr. Kathrin Nagele. And now I will play the pre-recorded lecture, and then we will have our discussion after. So bear with me 1 second while I get it running.
Thank you very much, Ashley, for that kind introduction, the invitation and the organization of this talk. And I, of course, also want to thank the audience today for your interest in our research. The topic today is ancient DNA and ancient genomics. And before jumping into the results that we have obtained through this method in the Caribbean, I'd like to briefly explain what DNA is, what ancient DNA is, and why it is so difficult to retrieve in tropical environments and how we managed in the end. So what is DNA? DNA is short for desoxyribonucleic acid, which is a chemical term describing the macromolecule that contains the kind of manual for all living creatures. It consists of a sugary backbone that is joined by phosphates and then has these nucleotides, which are kind of the letters of our genetic code. And of these letters, we have 4: guanine, cytosine, thymine, and adenine. And because DNA is not just there as a single strand, but as a double strand, they pair together. Guanine always with cytosine and thymine always with adenine, forming this DNA double strand. 3 of these nucleotides together always form a codon, and this codon defines a certain amino acid that, together with other amino acids, forms a chain. And this kind of chain of codons is called a gene. These genes produce chains of amino acids that fold and build proteins that are essential for human life. And this kind of double strand of DNA is not floating around in our body like this, but it's twirled into a double helix, which then again is densely packed onto proteins called histones, which in turn are kind of densified to coils that make up the chromosomes. And these chromosomes, of those we have 23 pairs. And one part of these 23 pairs is inherited by the father and the other half by the mother. So we can trace both parts of our ancestry with this type of DNA. Among those pairs, there's one special one called the sex chromosomes, where when the person is a female, most of them have X karyotype, which means that they have 2 of the larger chromosomes, and most males have one large and one smaller one, which we kind of denominate as XY karyotype. So this person here, for example, is a male person. The chromosomes, they are in the nucleus, so the core of our cells, which is why we refer to this DNA as the nuclear DNA. But this is not the only type of DNA that we have in our body. There is a second type that is coming from the mitochondria. So the mitochondria are most likely the most important organelles of our body, because in them, they create the energy that we need to fuel us, right? They are inherited only by the mother, and they contain a circular genome, which is why we call this genome the mitochondrial DNA, which is only inherited by the mother. But now what makes DNA ancient? Right after the death of a creature, the proteins in the cell start breaking down this macromolecule that our DNA is. And this is aided during the decompositional process by microbes that further digest this strand of DNA. So already shortly after the death of a person, the DNA is very fragmented. And this process continues over the years, decades, centuries, millennia to follow, until only very small fragments or nothing is left. And this process is accelerated by the presence of water. And it happens faster in warmer conditions. So one characteristic is the small fragment size, because water tends to take away the nucleotides, and on these positions, the strands break. And another type of damage is a chemical alteration of some nucleotides that's also aided by water. And with this, we kind of get the classic properties of ancient DNA, which are very short fragment lengths of around 45 to 50 nucleotides or base pairs. And also this chemical alteration that presents us with a very special feature that we call the smiley plot, because if we find like elevated amounts of these chemical alterations towards the ends of fragments, then we're happy, because that's ancient DNA. We can retrieve ancient DNA from a variety of sources, like fecal matter, cultural artifacts, hair, animal remains, plant remains, but most successful is the retrieval from skeletal elements. And with this new approach, with this new method, we've learned some very new and surprising things about the human past. For example, DNA from a finger bone of the size shown here from Denisova cave provided us with a new archaic human, the Denisovan, discovered in this cave, in the Denisova cave in Russia. And we learned that the spread of agriculture in Europe came with people coming from the Levant, in this region here, in what today is Syria and Israel, and not just by an innovation within Europe or by cultural kind of exchange between the groups, but with people. And we learned that from the ancient genome of Yersinia pestis, that the causative agent of the Black Death was in fact this bacterium that causes the plague. So this shows that we have a very great tool. It's a powerful tool to understand the human past. So of course, we also want to apply it to the Caribbean. But sadly, it's not that easy as in other regions, such as Europe, because DNA is a very complicated matter, and many variables factor into it. And of course, age is one very important variable. So the more time passes, the more fragmented the DNA will be. But age is not the only, and possibly not even the most important factor. We're only now starting to realize that the microclimate of a site has a very large effect, like for example, soil acidity or mineral content. For example, limestone is very beneficial for DNA preservation. But also the macroclimate is very important. And we've discussed before that water plays an important role in this degradation process. And in the tropics, it's very humid and it's also hot. And temperature is the other very obvious factor. This graph shows the average annual temperature in the world. And we have in like colder colors, the colder regions like here, Siberia, and then in warmer colors, the tropical regions. And it's no surprise that until 2016, the majority of ancient DNA sequences were recovered from human remains found in temperate to cold regions. But there is hope. One just has to use the combination of kind of the right methods. And ever since, we've developed new methods. And I just want to briefly introduce you to them. The first most important thing is to target the right element. And in the tropics, the two elements that are most promising are the petrous bone, which is this bone here. It contains the inner ear, so the cochlea, which we hear with, and the vestibular system that helps us keep our balance. And this bone is situated on the inside of our skull and is one of the most densest bones of our body. And those things together seem to be very beneficial for DNA preservation. And the other element are teeth, because the enamel encapsulates the tooth and also protects the DNA. Another measure that we take is to make sure that we do not contaminate the sample. So we produce everything in a clean room. And there we have to wear lots of protectional garment. And this is me shortly before the birth of my second son, wearing 3 pairs of gloves, a mask that we're now all familiar with, a hairnet, and this Tyvek suit to protect not me from the sample, but the sample from my DNA leaking from everywhere, my hair, my skin, everything. So we're trying to protect it. And dressed like this, we drill a hole most of the times into the element. This here is a long bone that we drill this hole into to produce this fine powder. And from this powder, this contains bone cells. And in these bone cells, we have the DNA that we want to. So this bone powder needs to be digested to open the cells and release the DNA. And then we have floating freely the fragments that were part of the sample. This is mostly environmental contamination, but also plant and pathogen DNA, and of course the DNA that we're interested in, the human DNA. Now, sequencing this cocktail can be extremely interesting because we can learn something about the environment in the site, about possibly ancient pathogens, what they suffered from. But it's also extremely expensive, because for every human read, for every human fragment that we want to read, we need to also read 100 to 1,000 other fragments that we're not interested in. And so what we've come up with is a method to kind of fish out the fragments that we're particularly interested in. And we do this with magnetic beads to which synthetic DNA molecules are bound. And these synthetic molecules are basically complementary to the DNA that we're interested in. So the DNA binds to those. And in this way, we can, since they're magnetic, we can keep them with a magnet and wash out all the rest. This can then be amplified or copied and then sequenced with next generation sequencing machines, which means that they can produce and read thousands and billions of these fragments within a very short period of time, making this much cheaper than it was before. And now, from this point, the computational work starts, because this machine now has the fragments and reads which nucleotide is in which position, and then provides us with so-called reads. And now these are like puzzle pieces, but of an extremely difficult puzzle, because we have billions of these puzzle pieces. And as I do with my children, when we do a tricky puzzle, I use a template so that I know which piece should go where. And this template we call reference genome. And it's basically a mixture of different human genomes from different places of the earth, and then all merged into kind of like a world human reference. And with this reference, the computer can now start and try to puzzle this back together. It tests on different places where does this fragment best fit. And once it found the best fit for each fragment, it provides us with a consensus sequence. And when we have a deviation from the consensus sequence from the reference genome, then we found something, a mutation that's called single nucleotide polymorphism. And those are the places in the genome that we're most interested in, because they are that because in our methods, we enrich for these single nucleotide polymorphisms that are that help us to distinguish the most between worldwide populations. And with this approach, then we can continue to contribute. So these are the sequences that ever since 2018 have been published. And now you can see that we are able to tell more about the human history of tropical places. And in fact, in the Caribbean today, we have more ancient genomes recovered than from the entire rest of the Americas. Which brings me basically to the place that I find most exciting, the Caribbean. And I will give you only a very short introduction because I do not want to carry owls to Athens. And you know this place better than I do. As you are all aware, the Caribbean is a place very much impacted by the European invasion in the 15th century. And as a result of the colonial crimes committed, the majority of Caribbean islanders today is a mixture of European, African, in some places South Asian, and First Peoples ancestry. The latter is represented only marginally in some populations and impacted by post-invasion displacements and migrations. For this reason, an attempt to study the ancient history of the Caribbean through present-day genomes, as is done in other places of the world, is too challenging. Another line of evidence often deployed are linguistics, but as another result of the invasion, also the linguistic diversity has completely changed and now is very much dominated by European languages. So the one field that has contributed the most to the understanding of the Caribbean before the European invasion is archaeology. And it tells us that the earliest occupational sites uncovered are in the extreme ends in the archipelago in Cuba and also Trinidad, but then are later also found at the center of the Caribbean, dating back to 7,000 to 5,000 years. This happened during the Archaic Age in the America, which is why the early settlers of this time are often referred to as Archaic Age settlers. I'm not a fan of that term because in other parts of the world, we use this for archaic humans, such as Neanderthal and Denisovans, and the settlers of the Caribbean do certainly have no connection to those. So that's why I prefer other terms. And since these groups show some variation in material culture, have a high cultural diversity, sometimes of showing lithic stone tools and sometimes of marine, made of marine resources, they practiced horticulturalism, so they changed actively their environment and managing their resources. They're sometimes called pre-ceramic, but we know today that they also had ceramic production. All of them lived a kind of semi-nomadic fisher hunter-gatherer lifestyle, which is why they sometimes referred to as fisher hunter-gatherers. The term that I like most is early settlers of the Western Caribbean, a term coined by a group of authors who proposed it in the book with the same title. The pattern of sites and associated dates led to various hypotheses of the origins of these early settlers of the Western Caribbean. Some believing South America or Central or even North America to be the region of origin for the first migrations. From 2800, the material culture changes starting from the south of the archipelago. We now find ornate pottery with regional variations, which is why this period is referred to as the ceramic age period. In addition, skeletons show artificial cranial deformation, and large-scale agriculture is the main subsistence strategy. It's not clear what happened before the advance into Cuba because there was a hiatus and whether the populations mixed, but the South American origin of this migration is supported by the first ancient DNA sequences, by archaeology, and also by linguistic analysis. What is contested though is which route they took. There is, we know also that there is a stylistic cultural diversity that perhaps pertains to cultural diversity, and this was also linked to different migrations from the mainland. It was certainly a diverse and pluralistic cultural scape, but how this kind of emerged is not clear. although the origins lie in the Orinoco River Delta. The southward expansion suggests that from the Orinoco River Delta, they traveled straight to Puerto Rico and then kind of back into the Lesser Antilles and further north, while the stepping stone mode suggests they used the Lesser Antilles as stepping stones in their advance into the Caribbean. There is a last dispersal that is very controversial, called the Carib expansion. And it started supposedly only 700 years ago. And it's not clear whether the Caribs were actually a people actually migrating from South America or a coalition of ceramic age groups from the Lesser Antilles who retreated from the Spaniards and formed a resistance. So there are various hypotheses towards this group of people that the colonizers called Caribs. So there are many open questions that DNA can answer, and some of them also pertain to regional and interregional interaction spheres that are revealed by archaeology, and also interactions between islands and the continents. Some of these questions we can answer with DNA. And in the early days, when all the methods that I've described before were not available yet, people used mitochondrial DNA. Because the mitochondria are so abundant in the cell and they have this ring-like structure, this DNA preserves better and is therefore recovered easier. The first studies conducted, they showed that in Hispaniola, there were C and D haplogroups. So these are specific mutations that you find on the mitochondrial genome and were kind of termed into different letters so that they would be easier to distinguish. And they found C and D haplogroups, which suggested a connection to South America. In Western Cuba, they found A, C, and D haplogroups and suggested South and perhaps also Central America. In Guadeloupe, the mitochondrial haplogroup diversity was very high, which they felt was perhaps a result of continuous contact with the mainland. And for the first time, they also reported the Y chromosomal haplogroup. So these are analyses of only the Y chromosome, which is only inherited by the father, and gives us an idea of the paternal lineage. Another study looked into the individuals from Red House, but I will go into detail later about the findings here. Now that we have the methods to also recover nuclear DNA, we can attempt to recover a more detailed picture and analyze not only the maternal and paternal lineages separately, but together. And up to now, we've already collected an impressive amount of ancient data. And in fact, as I said before, we have as many individuals now in the Caribbean than in the entire rest of the Americas. And I want to walk you through what we've learned from these studies looking into nuclear DNA. So in 2020, or since 2020, there were two very large studies published. This one is one that I was part of the team of authors, and it's drew also on previous research by my very dear colleague, Maria Nieves Colon, that you can see here, who looked into ancient DNA from Puerto Rico, and Hannes Schröder, who looked into the ancient DNA of one Bahamian individual. And the samples that we provided, this was a collaborative effort, especially with Cuban colleagues, where we recovered DNA from individuals that are associated with the earliest settlers of the Western Caribbean, and also from the Lesser Antilles from the Ceramic Age. And the first thing that we were interested in was to understand whether there were genetic differences between the two archaeological contexts. And we calculated genetic distance of each individual to all others. And then this results in this kind of clouds of individuals. So each of these points is one representing the genomic makeup of one individual. And the closer two individuals are in this space, the closer they're also genetically related. And what we see is, with some exceptions that we tested individually, that we have here a cloud of individuals associated with the early settlers of the Western Caribbean and individuals associated with the ceramic age. So we could show that they were genetically different. And after again showing that these are also genetically show an origin in northeastern South America, we were interested to understand in which way they entered the islands, whether it was a southward expansion or a stepping stone hypothesis. And so we built a tree, and kind of the succession of this tree would tell us something about how they dispersed over the islands. And so we looked what the branching pattern was, and we see that as we go up the chains, this is how they branch. So first we have a split of the St. Lucian individuals, then from Guadeloupe, and then we have Puerto Rico, finally the Bahamas and Cuba, meaning that this stepping stone is a better fit for the genetic data. We were also interested to find the origins of the archaic, of the early settlers of the Western Caribbean, so where they came from when they settled the Caribbean in the Archaic Age. But sadly, we were not able to do this because they showed a very generalized affinity to all people living, all indigenous populations of the Americas today. So what we did is we tried to model a tree with other ancient individuals from the Americas. And in this tree, we see that all Americans have a common ancestor in the Bering Strait. And entering the Americas, we see two ancestral lineages. These are called ancestral A and ancestral B. And B is represented only in North America today, and here by individuals from ancient South Ontario. Ancestral A contains ancient individuals from the California Channel Islands, but also all other South and Central Americans who show like a star-like radiation. And the ancient individuals from the Ceramic Age Caribbean are part of this radiation. Now, one individual in our set branched not with Central and South Americans, but earlier with ancient Californian islanders, revealing a second radiation event. When we looked at others that also dated to this period and some even contemporaneous, we see that they're not sufficiently modeled by coming from only this lineage, but that we need a mixture between this lineage and South American contribution. This does not mean that the origins of these individuals lie on the North American continent, because we cannot claim to have a good enough understanding of continental Americas at the time. But it shows that the Caribbean was settled and resettled multiple times already during the Archaic Age. And this suggests that there was repeated navigation, so that there was seafaring already during the Archaic Age. There was a second large study, and this study disagrees with the last finding. They argue that early settlers are most closely related to South American populations. And I'm afraid for now, we have to just accept that there are different interpretations of the same data. And hopefully in the future, when we have more data, this will become a bit clearer. This second study was focusing mostly on Ceramic Age contexts and closed a very important gap in our sampling, because before we did not have sequences from Hispaniola, although this is a very central island in the Caribbean. And what we learned, they also found one individual that dated to the Archaic Age. And this showed that was also genetically very similar to the Western Caribbean. So we learned that they also inhabited Hispaniola. And one individual from Diale 1 in Haiti was mixed with, of mixed ancestry with the newcomers also during the Ceramic Age, showing us that there was contact between the two. Although I find it very interesting that there is so little evidence for this contact, because everywhere else in the world, when two groups meet, they mix. And this seems not to be the case here. And potentially here we have another indication why during the kind of second settlement during the Ceramic Age of the Caribbean, there was maybe a halt because perhaps there was a cultural clash. We cannot know this through genetics. We can just observe that also genetically there's very little evidence for this mixture. They also looked into ceramic age inhabitants of Las Locas in Venezuela, so providing ancient genomic sequences also from the mainland. And they show that these are not the ancestors of the ceramic age inhabitants of the Lesser Antilles, but that the inhabitants of Curacao are a mixture between ancestors from Venezuela and from the Lesser Antilles. The most publicized, but at the same time most criticized finding, and I have to say that I also don't buy it, was the small census size. They claim that from the genetic similarities and because they identify close kin relationships within the Dominican Republic and between the Dominican Republic and Bahamas, they infer that the Caribbean was only populated by around 100,000 people at the time, not millions, as the colonizers' chroniclers report, and as also the archaeological record suggests. One finding, though, that all studies that have been done so far can agree on is that the ancient Caribbean islanders are not extinct, but they have left traces in the genomes of people today. And the first people's ancestry component of people in Puerto Rico today is most similar to the genomes of the ceramic age individual from the islands. And although genetics do not tell us something about identity, we can say that this efforts of reclaiming the indigenous heritage is also supported by the genetic analysis. And so we can say that there is genetic continuity with present-day islanders today. Lastly, a topic of interest in a context like the Caribbean are ancient pathogens. And in contact times, pathogens became very prominent. One theory is that indigenous population was naive to new pathogens and therefore had nothing to fight against the imported pathogens, suffering and dying from the illnesses such as the Kopelitzi, perhaps smallpox, syphilis. And we know that tuberculosis was present in the Americas before the European invasion, because from an ancient genome recovered from an ancient Peruvian mummy, we learned something about the evolution of this. Although this theory of kind of this naiveness of the first people's immune system is now partly refuted, because we know that there were diseases in the Americas before the European invasion. And today, many people believe that it has something to do with the results of the invasion, of like poor circumstances of living. One other pathogen, the very first viral pathogen that was studied is the hepatitis B virus that causes a liver disease. And today, strains from Europe, Africa, and Asia cluster together and show a worldwide distribution. Ancient genomes from Europe are most closely related to those in chimpanzees and gorillas, and the ones from the Americas seem to be ancestral to those. And this virus was also extracted from two 1,800-year-old individuals from Canimarabajo in Cuba, where these two individuals were also suffering from HBV. And the genomes recovered from them fell very close to those in the Americas today. But when we look at when the split was dated, it was dated to 7,500 to 5,000 years ago. Now I might go out on a limb a bit, but it might be that the evolution of this virus tells us something about the initial settlement of the Americas, because this date of the split coincides with what archaeology infers as the earliest settlement of the Caribbean. Now, let me finally summarize briefly the DNA work that was done and is still in progress that we do in Trinidad. You all know better than me, the Red House, the seat of parliament in the port of Spain. And during constructions in 2013, skeletons were discovered under the base of the building. And those human remains were studied by scholars published in a book in 2018. And it was found that they were part of ceramic age community that lived 1,800 to 550 years ago. A minimum of 60 individuals was recovered, and they showed signs of artificial cranial modifications, which is also observed in other individuals from a ceramic age context. More than half of the individuals showed signs of very poor health. And as part of this book, also the first DNA analysis was done, recovering mitochondrial DNA from a couple of individuals and showing that they had haplogroups A2, B2, C1b, C1d. So also a high diversity, very similar to Guadeloupe. And they also hypothesized connections to central and northern Venezuela and to the Lesser Antilles. These human remains are buried now in a ceremonial burial, but the samples taken were passed on to me, and I will try to use the described methods to recover the nuclear DNA. And in addition to the Red House, I was granted permission to extend these efforts also to Manzanilla, another ceramic age community. And the ceramic styles that were found at this site, they suggest connections to the Lesser Antilles. In a first screening, we tried to recover DNA from 17 individuals of each site. And here you can see the DNA preservation. So how much percent of DNA was preserved in the individuals. And for Red House, this is a very, very good preservation for a tropical environment. While for Manzanilla, this is more what we're used to. So it's not terrible, but it's also not something that I'm very happy about, because I was really hoping to understand this better. One of the reasons could be that from Red House, the samples that we looked into are from the petrous bone, while those from Manzanilla are from teeth. And now I'm very lucky that I was allowed to also sample petrous bones from Manzanilla, so that hopefully in the future, I will be able to present you more, because for now, only 4 of the 17 can be passed on to be enriched for human DNA, while we've done this for 14 from the Red House. And because this data has only become available very recently, I did not bring too many analyses, but I have a couple of preliminary analyses that I want to share with you today. For the Red House, I also did the same analysis that was done before about the mitochondria, with the very same results, really. So nothing too exciting there. But what I also did is I did a genetic sexing, which means I looked into which chromosomes, which type of sex chromosomes in each individual had, and I identified almost equal parts of females and males. And one analysis that I'm very excited about was I looked into the genetic relationship between the individuals, which can be indicative of like kinship relations. And I found 2 people, one male and one female, who were first degree related, which means that they were either siblings or father and daughter or mother and son. Earlier, I mentioned that analyzing the entire cocktail can be very interesting. And this is something that we also did. So we looked into what other types of organisms can we find. And this is kind of a graph summarizing it. I know it looks complicated, but let me walk you through it. So here we have in the columns the different types of microbes that we have. And in the lines, we have the different individuals up here from Manzanilla and down here from the Red House. Now, when we see something like this, we are always looking for kind of dark red colors, because they show us that this is most likely a positive hit of ancient fragments from this microbe that we have down here. When we see a smear like this, we can always suspect that it has to do something with the soil. And when we look into what this is Clostridium, for example, also a pathogen, but also a very common organism in soil. So what we see here is likely what is in the soil around the individuals. What we're more looking for are isolated incidents like this. And when we look into what microbe is associated with this, it's Streptococcus mutans. This is a bacterium that causes caries. which is something that we've known from the osteological analysis is very prominent in the site. So these two people, they definitely suffered also from caries in their mouth. And more excitingly is this positive fit here for one individual from the red house, because this shows that this person likely suffered from tuberculosis. And when carrying tuberculosis or being infected with tuberculosis, the immune system is suppressed, which would be an explanation for the generally poor health in the site if people were infected with that. Now, this is only a preliminary analysis, and now I will have to fish out the specific human reads also for Manzanilla, and then conduct a population genetic analysis, which means that I will compare this data to other ancient data from the Caribbean, and then be able to situate them and understand also their connections to other groups in the Lesser Antilles and on the Caribbean mainland, and understand something about the deep relationship within the site, but also between sites. And what is important to me in this process and the future analysis that I want to do is that it's a conversation. And I want to understand what's interesting for the people and indigenous communities in Trinidad, so that I can come up with analysis to test these questions and find methods to answer those. And I'm very happy that with Ashley, I have this very co-creative process of developing the questions. And while I'm thanking Ashley, I also want to extend this to the National Trust for supporting and facilitating this work, and more importantly, the exchange, to the Santa Rosa First Peoples community and the Varao Nation for their trust and endorsement of this work, and to my colleagues from the genetics institutes who have tried before me to recover DNA. And last but not least, to my wonderful boys and partner who allowed me to spend this time with you in Trinidad tonight and are very patient with me. And of course, I thank you for your attention.
Okay, thank you so much for that very informative talk, Kathrin. And I hope you're here with us. Kathrin, are you there?
Yes, I am. Thank you, Ashley.
Great, great. So remind me, what time is it in Leipzig right now?
It's 1:45 now.
Okay, 1:45. So thank you so much for being with us tonight and, giving us your time. So we would like to go straight into the discussion segment. We would like to hear your thoughts, your questions, both in the chat and via raise of hands. If you have questions that you'd like to ask directly to Kathrin, that would be fine. So not all at once, please start the question and answer segment. Thank you. All right, so let me see what's in the chat to start the ball rolling. So I see that you've been answering questions in the chat. Thank you so much for doing that. For all who did not know, that there are questions in the chat from a number of people that Kathrin has kindly answered. Let me see, are there any new ones? Okay, so I see this first question: did ancient pathogens from the Caribbean inhabitants also negatively affect the outside invaders and colonizers?
Thank you for that question. We think so, yes. We think that there are pathogens that originated in the Americas and that were, through the colonial contact and through the colonial invasion, even brought to Europe. Syphilis is one of those candidates, but also tuberculosis is suggested to have originated in the Americas. So we think, there's still much work to be done because we're only just starting to analyze ancient pathogens, both in the Americas and in Europe and other parts of the world, in fact. But for now, it very much seems so that this kind of went both ways.
Okay, thank you. All right, we have a question from Mr. McDowell. yes, a copy of this recording will be available, and no, you won't have to pay for it 1 red cent. You know, you can get that information from the National Trust website. It's either the website or it will be sent to you in an email, the link to the recording. So fear not, you know, the recording will be available to you. We're moving on now. Any questions that you'd like to ask Dr. Nagele directly? Anyone? Okay, we have a couple more. I see some hands. Mr. McDowell, you're first.
Yeah, I'm sorry if I was giving a video of my pocket earlier.
Okay.
Yeah, as I asked in the chat, you know, I know for a fact that my grandfather's mother, my maternal grandfather's mother, was partly native Trinidadian. And I did a DNA test with Ancestry. I can't remember, mydna.com. And what it showed is that I had a Native American connection. Well, I don't feel happy with that. You know, I'd like to have a connection that anchors me firmly in the Caribbean and in Trinidad and Tobago, if possible. And I'd like to know how is it possible for me to confirm that? Thank you very much.
It depends on which provider you use, because some offer also tools that allow you to analyze your DNA with other ancient sequences. And I'm not sure, maybe we can make the ancient sequences from the Caribbean available to them because they're openly available. So you could, in theory, just go and download them and look for yourself. But I understand that might be tricky. So some providers, they offer this to co-analyze your data also with ancient sequences, or there are even some tools online.
Okay. Thank you, Joseph. I see that Neil Phillip asked a question in the chat, but your hand is up, so you would like to ask it live?
Well, I had another question in addition to that, so I don't know if it might be better to read the first question first and then I'll ask this one.
Okay, no problem. The first question is the presence of DNA reads from pathogens or other organisms always indicative of the disease state? If not, how does one infer whether the disease state was present or not?
Thank you for the question. I'm not sure that I understand it correctly because I'm not a native English speaker. Does disease in this case mean that the person had the disease or that the person died?
Just if they had the disease.
Okay. I would say that it is a very strong indication that this person suffered from the disease, because otherwise the viral DNA would not be present in such high numbers in their bloodstream. Which is why when we detect the disease, we can be almost certain. Scientists never say we're 100% certain. We're almost certain that this person did suffer from the disease. The contrary is actually the more obvious, or not obvious, the more common, sorry, getting late here, my English is getting worse. The more common state is the contrary, that we know that people suffer from a disease. Like, for example, in medieval Europe, we have mass graves where it's recorded in books that all these people died from the plague. But when we test every single skeleton that is in this mass graves, we only find it in 1 or maybe 2. So we know that these people very likely died from the plague, but we can find the DNA only in 2. And so recovering the pathogen's DNA is still very difficult. And when it's there, we are very sure that this individual suffered from the disease. Does that answer your question?
Yes, it does.
Okay, thank you so much, Mr. Phillip. We can move on to a question about Banwari man, or we now know it's Banwari woman. The individual was in fact a woman. So I'm not sure if you're familiar, Kathrin, with, yeah, you're familiar, great.
Yeah, I am.
So great. So there's Banwari woman, the oldest Trinidadian that is in the Zoological Museum at the University of the West Indies. What would be interesting to add to our study is, okay, so she's asking about the Banwari man, and if you plan the Banwari specimen, and if you plan to study that individual, or, you know, that kind of.
It would certainly be amazing to be allowed to study this individual, because it is so far the oldest individual from Trinidad and from the southern Caribbean, because we have very old individuals also in the north, in Cuba, for example. But it would give us an indication of what the archaic age in the south looked like, and specifically in Trinidad. But this is a very unique individual. So, and our methods are very often destructive, or let's say that with the destructive methods, we get the best results. So there is something to balance here. On the one hand, we have this very unique individual, the very unique samples that we have, and we don't want to destroy them, right? But on the other hand, we want to also understand their DNA. And so for now, I've proposed several ways to sample this individual, and now it's up to the authorities to decide if they want to go ahead with this now, or if we rather should wait until perhaps better methods become available in the future.
Mm-hmm. Well, I'm sure that there's a conversation to be had there, and maybe we will look into that a little bit after this presentation. Okay, so moving on. Let me just ask, is there anybody who would like to ask a question live before we move on? Going once, going twice, not so. Okay.
Yes.
So moving on.
Hello.
All right, Lancelot. Hello.
So my question is to Dr. Nagele. Now, I have learned there's a term, the Columbian exchange. When the early Europeans, namely the Spanish, came in the later part of the 15th century, there was a migration of pathogens. It is alleged the Europeans brought the European pathogens, and they came into contact with the early indigenous people, and they took some of the indigenous pathogens back to Europe. Could Dr. you shed some light on this, please, on those pathogens in the Columbian exchange?
So we think that pathogens that traveled back with the colonizers to Europe could be, for example, tuberculosis, which became a big pandemic in Europe afterwards, and also syphilis, which in the colonial period, many people in Europe suffered from. Another indication, like during the colonial period, there was lots of travel also by pathogens, or they traveled as part of, for example, the transatlantic slave trade also brought strains of certain pathogens to the Americas, which from three individuals that likely represent enslaved people from Mexico. They were brought there from somewhere in Western Africa, and they also showed pathogens that had that were most closely related to the same type of pathogens that we see in West Africa today. But I would have to check which ones exactly those were.
Thank you. Okay, so moving on, we have a question, an interesting question. Is the ancient DNA history of Trinidad biased towards the Carib peoples, in quotes, the Carib peoples, as opposed to other groups in Trinidad?
So for now, regarding this Carib migration, there is no genetic evidence of Caribs in the island so far. I mean, we've not extensively sampled the Lesser Antilles. There's still room for this to be discovered, but for now, even from sites that have been discussed as Carib sites before, they look very much the same as other ceramic age groups do. So there is nothing genetically different. I mean, we cannot speak for identity, we cannot speak for culture. So there is still room for this, and we certainly do not want to kind of impose this ideas of or terminology on people living today, right? So people today identify as Caribs, that's their right because it's their culture and their identity. What we look into are the ancient peoples that cannot speak for themselves anymore. And there we cannot find a genetic difference. So for now, when it comes to the ancient Caribs, we haven't found them yet.
Okay. But hopefully further research will shed light on that issue. Okay, so we have Darlene Anderson with her hand up. Darlene, could you ask a question, please?
Yes, hi everyone. My question is, I actually wrote it down, but my grandmother was actually one of the volunteers for the University of Puerto Rico, where they studied her DNA for native markers. And in her DNA and also in mine, it reveals that we have Native American Taino and Native American from the Mexican Peninsula. There's one finding in a cave in Puerto Rico that has a Mayan calendar. So do you have like any information as to the connection between either natives from the Mexican Peninsula with the natives from the Caribbean?
Thank you for the question. So far, we don't have genetic evidence for this contact, but there are plenty of indications in the archaeology that there was very busy traveling back and forth between the islands and the mainland. But as I said before, but it was more like a side note, our problem is that in the Circum-Caribbean, we don't have many ancient individuals to compare to. And since the colonial invasion has impacted this region so drastically, it's very difficult to use modern genomes to do this, right? So even if we looked into Mayans today and compared it with the ancient islanders from the Caribbean, we might have a bias that's introduced through the colonial invasion and kind of the restructuring of the entire continent. And this is why before we don't have the ancient genomes also from the Circum-Caribbean, it's very difficult to answer these questions with DNA. But it's certainly something that we're very interested in and working on.
Thank you.
Okay, thank you for your question. Moving forward, we have a couple in the chat, many in the chat actually. We'll start with Kathrin. Kathrin asks about if you've looked into the African regions and their representation in Trinidadian DNA.
Sadly, I haven't, because my expertise is with ancient individuals, and I sometimes look into modern humans or like presently living humans when it's interesting to answer questions of the past, like for example about continuity. I know that many people have done this on other islands, like for example in Cuba or Puerto Rico, and found that mostly this is from West African origin. I don't know how well it can be traced to a certain country, although this can be attempted. And since the work in Trinidad is only just beginning, I haven't even checked if there is data available from Trinidad to do these analyses. But now that this is picking up and we see that there is DNA preservation, which before we didn't have, now we can try and assemble these data sets and potentially also look into that. Yes.
Okay. One more from the chat. Bearing in mind the pathogens, does your work extend to pharmacopoeia of ancient peoples? This is from Maria Gazelle Mason Roberts.
I have to say that I never thought about it, and it's certainly not work that I would be able to do, but I have a colleague and she looks into dental calculus. So I don't know how familiar everyone is with the term. I had to learn it. So the stuff that's on your teeth that the dentist scrapes away, that's calculus. And we find this on ancient teeth a lot. And this plaque, or this kind of fossilized plaque, incorporates everything that you eat. So, and I could imagine that pharmaceuticals is something that, you know, you would maybe administer orally, for example, and then it might catch in, or how do you say this? It might get caught in the calculus. So this would be something that this colleague who's studying the calculus could potentially look at. The traces of the medicinal plants would not show up in our ancient DNA analysis, because that would be something that's in the bloodstream and then only in very small amounts, so that it's probably not traceable.
Okay, interesting. Okay, so live we have Mr. Shamsuddin, which we're very happy to have in attendance, because he is a genealogist of some renowned in Trinidad. So genealogists, geneticists walk into about, I don't know what they say, but this is interesting to hear. So Mr. Shamsuddin, please. Hello? Are you still with us? Okay, we'll give him some time. We'll move on to another question. For now, we also have, oh, I think he's here.
Okay.
Yes, I'm hearing you now.
Yeah, sorry, I was getting difficulty unmuting myself.
No problem.
I just want to come back on the Banwari question.
Yes.
I know that a whole group of people, a new race almost of people, I don't know if that's a strand of human being, the Denisovans, were found by just the remains of a finger, I think. Could we not take a little piece of Banwari, not destroying all of him? Could we not take a bit of him and get some more details of this ancient DNA and see where we stand on this? Is that a possibility or not?
That would be ideal. And we usually, it was maybe misleading to say that it's very destructive. We don't destroy entire skeletons, but we have to chip off pieces. And then it's not so nice to exhibit them in the museum anymore if they're like pieces broken out of them, which is why I'm hesitant to just saw off a part of the tooth, for example, that we have from Banwari trace. And this was achieved, yes, in Russia. And it's important to mention because there, this cave, so that's the first thing, it was a cave, which protects very well and which provides a good microclimate that's always cool. And so this is very good for DNA preservation. And then in general, the climate in this region of Russia is very stable. So the preservation there is really ideal. Like even from the soil in this cave, you get DNA. In the Caribbean, the situation is entirely different. There we really have to make a headstand and wiggle with our toes to kind of get some DNA from the elements that we have. And so I'm afraid that like very, very tiny pieces will not cut it. And this is also why I'm hesitant, because I feel that it's not worth destroying the sample from this individual, and then we won't get any DNA out of it. So I first would like to make sure that we can get consistently good DNA preservation from different sites across Trinidad, from different time periods even, approaching the one of the Banwari trace. And if we still can recover DNA from that, then it's really worth trying. And I mean, if I'm granted the permission to try it, and if people are happy with me to do it, then I certainly will, and I will do my very best.
Could I follow up one follow-up, please?
Sure.
Yeah. Sure. I was just wondering the difficulty of getting DNA from tropical areas like ours. Could be of course because of the conditions that you mentioned, the heat and the rain and so on. Is it possible, hypothetically speaking now, that we may have to think of the coming of humans to the new world to be even further back than we know it at the present time, or we've read about it? Is it possible that it could have been much earlier? Because we're now knowing that the Bering Strait may not have been the only means by which they came. We are hearing that they could have come otherwise, Chile and so on. We are hearing human beings there even around the time or before the Bering Strait. So is it possible then for having made it to the Caribbean area or to the tropical areas of the New World, that their DNA could have been destroyed or still very, very difficult to find? Is that a possibility at all, or is it only hypothetically speaking?
I think that is a possibility, but this is something that geneticists in the Americas fight over a lot. And there is now mounting evidence that this landway through, like waiting for the glaciers in North America to melt, for people to come into the Americas, we already know that this was not the case, because these glaciers melted after we have the first occupational sites in South America. And in the beginning, this was kind of contested because there was only this one site and everyone thought, oh, this person made a mistake, it cannot be that old. But now we have multiple sites that predate the melting of the glaciers. So it's more and more, like we are growing, or many people are, this idea is growing on them, that they were people before. And now there were footsteps found, preserved in the dirt in, I believe it was New Mexico, that date to around 20,000 years ago. And it was clearly humans walking there. And of course, the researchers there also made sure that they had kind of bulletproof evidence for this date. And I'm very much inclined to believe it. Another factor is that in South America, especially in the Amazon, we find a signature, a genetic signature that relates, or that part of the genome is related to people of Oceania, which was dubbed the population Y signal. And this has popped up every now and then in isolated populations that live today. But now we've also recovered it from ancient genomes. And ever since, I think that this population Y signal might be something that is a kind of a leftover of this very first migration into the Americas, possibly much earlier, yes. But this is something that we do not have solid evidence, because for now we have not retrieved DNA from 20,000-year-old human remains from the Americas, because they're not preserved and we haven't found them yet. But the most likely place to look for them would be the Amazon, which is a very difficult environment to work in and to find sites, occupational sites. It's easy when there are structures, big structures, like for example, the Mayan mounts, or like Mayan structures, they can be found with surface radars. But when we look into earlier communities that did not build massive structures, then it becomes difficult to find these, and you would have to kind of find them in the jungle somehow. And I'm sure that this is difficult for any researcher to do. So we'll have to wait for incidental findings, and then hopefully they will give us DNA with better methods.
Yes, hopefully. Okay, so we're kind of coming up to the limit, but we'll try to answer as many questions as we can. Moving on. Thank you, Mr. Dean. Okay, so from Bridget Brereton, who's a noted historian as well. The scholars who came up with a very low estimate of the population of the pre-contact Caribbean, how did they react? How did they reach that estimate? And why do you disagree?
So what they did is they looked into tracks of the DNA. So they compared in different individuals, how big are the tracks that they share. And the bigger the tracks are, the more related your parents were, for example. And by kind of understanding the frequency of certain sizes of these tracks, one can estimate how much consanguinity was, like background consanguinity was in the Caribbean, which means that there was not a lot of people to mate with each other to keep the population alive, let's say. And this is how they identified of certain tracks of certain sizes, and then concluded that the population could not have been that big. So they did some simulations and looked also into other regions of the world where this might be known. And this is how they kind of came up with this number. The reason why I don't buy it is because of their sampling. So they looked into very specific groups on Hispaniola and also in the Bahamas. And I think that there might be a bias, one, because they looked on it only on the sex chromosomes. And this is tricky because they are, inherited by the father and by the mother. And although there is some variation and some inheritance patterns, they still don't represent it as well as all the other, like taking into account all the other 22 chromosomes. And the person who has developed this method is currently working on developing the method further to also look into the autosomal chromosomes. And I'm very interested to see what he will find if we look at the entire genomic DNA that's available. And then I would also like to see this extended to other places in the Caribbean and kind of recalculated based on the entire set of samples and not only from Hispaniola. Because I think that there might have been a bias introduced, that they were looking into one interaction sphere where we see that there is also trade been going on. So maybe this community trading were 100,000 people, but maybe there were other communities trading with other places in the Caribbean with another 100,000 people and another 100,000, and summing up those kind of trading communities and interaction spheres, we would get to a much higher number. That would be my prediction.
Mm-hmm. Thank you for that. Hopefully, Professor Brereton, that answers your question, and I'm sure Dr. Nagele will have her email address in the chat, so people can ask further questions. So let me do a little lightning round here. I mean, I have 25 messages that I haven't even looked at yet, and so maybe we should have allotted more time for this. It's, people are very interested in asking you some questions relating to your research. So let's move on very quickly. Okay, I can answer this question. Akila Stewart is asking about, well, she didn't know that there were ancient peoples from Manzanilla. And so the detail I can share is, archaeological discoveries have happened all over Trinidad. We have over 300 archaeological sites. That doesn't mean that all of these sites have produced human remains, but just in Manzanilla, we have about 3 known sites. I think there are a couple more, but to my knowledge, there are about 3 known sites in Manzanilla to date, and I'm sure there are many more because of the interaction that happened on that coast. So I think that, yeah, we have a lot of information from Manzanilla. I can probably direct you to some literature from Dr. Ari Boomert. He has a book, gosh, I forget the full title right now. But if you search Dr. Ari Boomert, you'll find quite a number of publications based on Trinidad. So you'll see a little bit more about the archaeology of Trinidad in those books. So moving on, we have, will foreign, example, US and UK DNA tests be accurate to match persons residing in the Caribbean who have been here for several generations? Kathrin, I can't hear you. Are you on mute?
Yeah, I was asking for you to repeat the question. I couldn't find it in the chat and I didn't understand very well. I'm sorry.
Sure. Will foreign, example, United States and United Kingdom DNA tests be accurate to match persons residing in the Caribbean who have been here for several generations?
I'm inclined to say yes, because the data sets used are worldwide data sets, and the genetic markers that they're looking for are some that are very good for not discriminating, what's the word, for identifying differences in worldwide populations. So it doesn't really, it's not a problem where these tests have been designed, but more the data sets that go into it. And that might be a problem when only people of a certain country are asking to be sequenced from this provider, because with every customer, they're kind of increasing their data set and they're refining their methods. So that might be something to look in, where, from which part of the world people are mostly sending in their DNA to a certain provider.
Okay.
But in general, the methods that they use are the same methods that we, for example, also use to determine this. And for this, we use a reference data set that is publicly available also to them. So they won't do a better or worse job, let's say. They might not run as many iterations, so they might not try to get very, very close to the truth, but close enough, I would say.
Okay, thank you. All right, just before we go to Ms. Manuel, let me just do one more question from the chat. What is the general level of confidence attributed to the computational sequencing of the fragments of ancient DNA? Also, do you believe that other persons from other periods / types of persons exist other than those already known? I'm not quite sure about the second part, but if you can answer the first one.
I'm not sure about the first one, if I understand correctly. So the confidence that we have, so when these fragments are sequenced, then the sequencing machines, they give us already an indication, like a score of how well they were able to read a certain position. And sometimes the score is bad, but mostly with this new machines, and in our institute we have very new machines, we can be very confident that they have read this fragment in the right way. And then also in the computational process, there are other quality scores, let's say, that tell us how well did it map to a certain reference, how well does it fit in this position in the reference. And we exclude reads, so these fragments that do not pass a certain threshold of quality. So in the end of this consensus sequence that we're creating in the end, this is a very high quality. So with this, we're very confident that what we have is the actual sequence of this ancient individual. And I hope this answers the question. And the second part, the way that I understand the question is, if I believe that there are kind of, we sometimes call them ghost populations. So populations that we know should be somewhere, but we haven't found them yet. And I mentioned one of those ghost populations earlier as the population Y. This is one of those ghost populations that we see contributions of this population that is not known to us in certain groups, like for example, in the Amazon. We also see kind of ghost archaic hominins. For example, in Asia, we sometimes have groups where there is contribution that doesn't fit other humans, that doesn't fit other archaic humans that we know. So we believe that this must have been like introgression of an unknown archaic hominin. So these do exist, and I hope that I understood the question right, and that this was the information that you were looking for, Paul.
Okay. So Alison, you have your hand raised. Can you ask your question now, please?
Yes, good afternoon. Thank you for giving me a chance to ask. I'm just a regular layperson, so a lot of this, with the genetic stuff is a little bit above my head, but I did a DNA test, hoping to kind of clarify, of course, my ancestry. I am from Trinidad. My parents are from Trinidad and so forth. But instead of kind of clarifying, it's sort of opened up a little bit more because of course, I was expecting the African ancestry, but then it's opened up to a lot of indigenous, but from different areas. Like I did it on Ancestry. So I'm seeing indigenous from Yucatan, and then from Puerto Rico, Bolivia and Peru, even Levant, Americas, Panama, Costa Rica, indigenous Cuba. So it seems that, and also Eastern South America. So it's kind of made me even more confused now because it seems like somehow my ancestry is just spread out all over. And I know she was talking about trying to figure out the patterns of migration. But like the thing is, I don't even know how to interpret this for myself. And my second question is also, can we as people indigenous to Trinidad, can we be somehow participatory in the research in some way?
So to answer the second question first, yes, I think so. We have to go through ethical clearance for this, and there has to be like a hypothesis that we are allowed to sample DNA from today living inhabitants of Trinidad. And if we feel in this project, and we'll discuss this also with the Trinidadian colleagues, that this would be beneficial for us to understand the history of Trinidad better, then this is certainly some hoops that we are willing to jump through to get this ethical clearance, and then to come to the island and sample DNA. But this would be something that it's not like the ancestry test where you send us a swab, but we would have to come and explain everything and explain also what the research goals are and so on. But in theory, yes, this would be possible. And for your first question, I've never done an ancestry test myself because I believe that for me, it would be extremely boring. My ancestors are farmers in southern Germany since, I don't know, as far as they can think back. So I'm pretty sure that this would be my ancestry. And I understand that in the Americas, it's more exciting, but also more confusing. And I mean, of course, you said yourself you were expecting the African component because, well, we know about the MAFA or the slave trade and the transatlantic slave trade. And now you're wondering about this kind of mix that you have of Native American ancestry, if I understand you correctly. And there are multiple explanations, some from the pre-colonial times, some from the colonial times. So in the pre-colonial times, there was exchange between the islands. So people of Trinidad most likely also were in contact with other people from the Lesser Antilles or even from Puerto Rico, from Venezuela, from other places nearby and far away. When we see something like contributions from Central America or from Yucatan, then this is most likely the displacement of indigenous people during the colonial times, when people on the islands died and the colonizers were in need of more workforce. They not only brought them from Africa, but also from the continent to the island. So this massive displacement was not only across the Atlantic Ocean, but also across the Caribbean Sea. And this, I would suggest, or I would interpret it as kind of traces also of this displacement.
Thank you.
Okay, thank you so much. We've come to the end of our discussion segment. As I said, if you have any other burning questions, you could ask Dr. Nagele yourself via email. And I think she's going to put her email again. Yes, great. Thank you so much. This has been a very, very thought-provoking program. And I thank you for your lecture. And I hope we can probably do this again at the end of the research, if it does have an end. But thank you so much. I'd like to take this opportunity now to thank the National Trust for having this event. Also, for all other persons behind the scenes that work to make this event a possibility. And of course, for the patrons, thank you so much, audience, for all your questions and your attention and your time. Thank you so much. Just a little housekeeping before we go. I have to tell you about the tours that we have scheduled for the month of June, because the National Trust, yes, we engage in lectures and so forth, but we also do tours as part of our outreach program. So we have 6 tours carded for the month of June. The first is Paradise Cemetery Tour, followed by Erin Heritage Tour, Erin Adventure Tour, Faizabad Heritage Tour, Chakachakari Tour Heritage Tour, Alabre Adventure Tour, and I think that's it. So if you want to learn more about our tour offering, you can go to our website, and I'm sure it's in our in the emails that we send out to membership. And speaking of membership, in order to make events like this possible, we really rely upon our membership. So we thank you so much for those who are already members, and we hope that people who are here that are not members will consider joining the National Trust on our journey. So again, thank you so much. Thank you, Kathrin, for being up so late. And I hope to see everyone soon. Thanks again.
Thank you very much for inviting me.
Okay. And one last thing, the recording of this program will be available in probably maybe a couple of days, and we will publicize that via email or on our website. So thanks again for everyone for coming, and we'll see you next time. Thank you.